Okay, so we'll continue the next section with Professor Djebli from Algeria. Please, Professor, tell us about your experience.
Okay, thank you, Professor Stefan. Donc, good morning or good afternoon, everybody. I introduce myself. I am Professor Noureddine Djebli from Mostaganem University, Algeria. First of all, I would like to thank Professor Stefan and Professor Nariman for their invitation. So today I'll try to present a part of my PhD student work. It's about evaluation of antidiabetic and healing honey from southern Algeria in vivo study. Okay, about diabetes, its world prevalence continues to increase. In 2019, the number of adults 20 to 79 old with diabetes was estimated to be around than more for million people, representing 9.43%. By 2030, the number is expected to increase to more to 5 million people. And into 2045, it's estimated to arrive at 7 million people. I think it's very, very serious problem in the world. So in 2019, by International Diabetes Federation estimated that 40 million adults aged 20 to 99 died by this pathology. Sorry. Okay, what is the complication of this pathology? Diabetes is one of the most important comorbidity linked to severity SARS-CoV-2. Early data to emerge from Wuhan in China showed a prevalence of diabetes of 12 to 22% among patients hospitalized with COVID-19. I think it's very, very serious problem. About the different factor of this pathology, if, as you know, it's genetic, the environment, in the two cases, type 1 or type 2. Now, what is about the treatment? We have the synthetic treatment. It's about insulin, as you know. Insulin is the main treatment for type 1 diabetes. Insulin, secretagogue is the class of drug including sulfonyl and methylglendine. It's the second classes of synthetic medicament to treat diabetes. But I think it's have the other problem in the human organics. For example, if we say what is the problem of talasamide or acetohexamide, I think it's the problem. About natural products, I mean medicinal plant, we can treat this problem, diabetes, by different medicinal plants, as for example, Punica granatum, and we have published in this research that we found that Punica granatum, it's very, very interesting to treat this pathology, I mean diabetic. Now, what is the role of epitherapy? If you know, epitherapy is the study of using bee products such as honey, pollen, propolis, etc. to treat many different pathologic or disease as diabetics. Now, what about honey? Honey is natural product produced by both honeybees and stingless bees. Both types of honey contain unique and distinct type of phenolic flavonoid compounds, variable biological and clinical importance. Honey is one of the most effective natural products used for wound healing. About different therapeutic of honey, if you know the traditional use and clinical application of both honey, such as antimicrobial, antioxidant, anti-inflammatory, antioxidant, etc. Wound healing activity are described. In the first activity about antimicrobial, before 7 or 8 years, we worked on the different activity or on different antimicrobial activities that we publish more 20 paper in this area that we found every time that honey, and which honey? I mean honey of cedar. Okay, it's very, very important to in these activities. I mean antimicrobial in general, antibacterial, antifungal, antiviral, etc. This is some paper for this area that my PhD student before were 8 years worked on it. Today I try to present some works of my PhD students that the aims or objective of this work is included the evaluation of the antidiabetic and healing honey from southern Algeria in vivo study. Okay, the first step of this work that we collected honey from steps location in Algeria. I think that Nariman know this part of our country. Our country, Algeria, it's the bigger country in the Africa, and we are, I think we are very, very lucky because we have 4 seasons in the year. And also we are Mediterranean country. Okay, the collection of honey was from location of Jelfa, exactly in Hassi Bahbah. This is one city of the steps location in Algeria. What is in the material methods, we have 2 parts, in vitro study and we have in vivo study. About in vitro study, we measure different physical-chemical analysis and different physical chemicals, I mean water content, optical rotation, pH, and proline, spectrum of sugar, etc. And we measure also phenolic compounds by HPLC. Also, we measure different secondary compounds as polyphenols and flavonoids. And also we try to measure antioxidant activities as DPPH and FRAP by this different method. Okay, now we pass in the in vivo study. In vivo study, in the first time, we have to pass by toxicity test of honey. The honey was administered orally to rats. We have 3 groups. The first receives the honey solution at rat of 250 milligram per kg. The second groups rat of 500 milligram per kg, and the third, the last one, receive a honey solution of 1 gram by kg. If you see, we have 3 groups to test of toxicity test. We do it by this reference OECD, and we continue to see carefully and monitor it and observe it for the first 4 hours and daily thereafter for 40 days. The duration is 40 days to see what about this product, I mean honey by different concentration. Okay, now about antidiabetics. If you see, antidiabetics, we have 6 groups of animals. We worked on rats. We have 6 groups. We have control, and we used metformin as synthetic treatment of a diabetic at 33 milligram by kg, and we have 3 doses of cedar honey, 250, 500, and 1 gram by kg, and we have in the last this is the diabetic control. Okay. In the first step, we provocate diabetic by STZ, if you see, streptozothin at 6.5 milligram per kg by intraperitoneal for 2 weeks. After 2 weeks, we treat the rat by these different doses by honey, and by this metformin as treatment, synthetic treatment. And after 2 weeks, we pass to take the blood and the pancreas for histological study. About the second part of healing honey. It's very complicated, but we arrive to do it. 1 day before the experiment, the rats were shaved, if you see this one, and they are anesthesied with ketamine and lidocaine. The second step, that after cleaning the shaved part with the surgical calhoun, 2 square, if you see, this is the first and this is the second. 2 square are drawn for each rat. And in this step, we provocate wounds, skins removed. And in this step, we treat by Madicasol, a synthetic medicament, and we do application of honey, topical application of honey. In the last step, we covered the wound with bond gases. This experiment, it's the duration for this experiment is 13 days, 1-3, I mean. Okay, about the results, we have physiochemical parameters of cedar honey. If you see acidity and optical rotation, in general, we can estimate or suggest, sorry, that our sample of cedar honey, it's very, very interesting, and it's very, very important as a natural product. This phenolic profile that we found that our honey, it's very, very, very rich about, if you see, about sorry for phenolic compounds. I mean, for example, acid gallic, acid syringic, etc. What about now? What about antioxidant activity? If you see that this value confirm that our sample of honey, I mean cedar honey, it's very active to stress antioxidant activity. Now about the toxicity test. No sign of toxicity in rat was noted after administration of the different doses of honey. Okay, 250, 50, and 500 and 1 gram by kg for 40 days of monitoring. Okay, now what about anti-diabetic activity? I mean in vivo study. The first one that we try to measure the evolution of blood sugar. If you see, we have 3 parts, 2 weeks and 4 weeks and 2 weeks. 2 weeks, it's after provocation diabetic. And 4 weeks, it's for treatment. And 2 weeks, this is our hypothesis, that we can continue to observe it, what is the value of blood sugar. If you see, this is the control, okay, and this is the diabetics, and the treatment is between diabetics and between control and diabetics. We confirm that it's a very good activity anti-diabetic of our sample honey. Now this is the weight body. If you know, this is the control, and this is the diabetic, and between this and this is different doses of honey. Okay, other parameters. I mean, for example, biochemical parameters to confirm if we have interaction between diabetics and lipidic parameters. If you see, we measure this is the cholesterol, triglyceride, HDL, and azat and alat. This is the hepatic profile. We confirm that we are correlation and positive correlation. In the second, we try to measure other biochemical parameters as urea, creatinine, and some mineral as potassium, sodium, and chlor. Why? Because the problem that's the consummation. If you see the consummation of water, it's very important in diabetic. We continues to see what is the different histopathology study, in particular in pancreas. If you see, this is Langerhans eye. It's very clear in the control, and if we compare it this one by in the diabetics, if you see, this is different damage destruction in the Langerhans eye, and this is for the treatment. We have different doses, if you see, and we have synthetic medicaments. If you see in these doses, for example, or in this, the structure, it's approach to control groups. If you see, this is the island Langerhans, sorry, and this is the same when we use 250 or 1 gram. Now, what about the healing activity? The healing activity, we try to compare between two big groups. We have diabetic rats and no diabetic rats. In the first one, in diabetic rats, if you see, for example, this is diabetics control. We after 10 days, we saw that the problem of healing it's positive comparative the 8th and the 6th days. This is when we use decamazole. We see that in the 10 days, no healing. We can now compare this one by this one. In this one, we use cedar honey, and I remind you that we worked in diabetics rats. I mean that we provocate the wound in groups of diabetics rats. Other results. This is the concentration of wound healing during this period of experiment. If you say, for example, this is honey in this color. If you see, okay, that we confirm that it's very good product to treat this problem of wound. In this picture, that we measure the percent of contraction. I mean the contraction of the wound in diabetic rat. If you see, for example, this is diabetic control in this color. Okay, it's similar if when we compare it to honey and synthetic medicament. But in the second groups, I mean no diabetic rats, it's similar results. If you see, this is the control, and this is by synthetic treatment, and this is by honey cedar. If you see in this one or this one or this one, after 10 years, we can say that we have the good or the very good treatment when we treat our rats by honey. Same data when we measure the percent of contraction. We confirm that the treatment by honey, it's a very good product to treat this problem. And the second one is to confirm the prevent data that we have similar data between cedar honey and the control. This is in general some data for the work of my PhD student, that we confirm that our sample from steps location, I mean cedar honey, it's very interesting. And sorry if I was very fast because problem the screen. In the conclusion, I can said that different physicochemical characteristic meet standard, okay, and it's similar to Codex Alimentary, very rich of polyphenol, flavonoid, that we when we used 13 standards as resveratrol, creatine, and others. Our sample can reduce the blood sugar, okay, and improve the weight, cholesterol, and other parameters. And it's very good healing with very rapid evolution and it was significant anti-ulcer activity. Now I present some picture of my city. My city is Mostaganem. It's we have the longer beach in my country, in Algeria, and this is the monument of the period of Ottoman in Mostaganem, and this is the new city of my new building, sorry, of my city. This is my laboratory. Laboratory is pharmacology EP phytotherapy laboratory. Laboratoire de pharmacology EP phytotherapy. This is souvenir picture of my team, PhD students, old one and new and a new one. And finally, I can see. Thank you for your attention.